Hi friends, welcome back to my channel. So if you're new to my channel, please kindly like, share and subscribe to my channel. Okay, Tackleknowledgechannel. So today we will be starting new textual book. Okay, Cell and Molecular Biology Concepts and Experiments, Geraldkarp, 7th edition. Okay, so we are going to start with this textbook, first chapter. Okay, so in this textbook we will be first starting with the chapter 1, okay, Introduction to the Study of Cell and Molecular Biology. So in this we will be seeing the discovery of the cells, basic properties of cells, two fundamental different classes of cells, viruses, the human perspective, the prospects of cell replacement therapy, experimental pathways, the origin of eukaryotic cells. So all this we are going to see in this chapter. Okay, let's begin with the chapter. So first, cells and the structures they comprise are too small to be directly seen, heard or touched. So if we see the cell and its structures, they are comprising too small. Okay, so they are comprises with the too small, which are directly seen. Okay, they can be heard and they can be touched. Okay, in spite of this tremendous handicap, cells are the subject of hundreds of thousands of publications each year, with virtually every aspect of their minuscule structure coming under scrutiny. So there are tremendous handicaps. Cells are subjected hundreds of thousands of publications each year. Okay, so with virtually every aspect of minuscule structure, they are under scrutiny. Okay, in many ways, the study of the cell and molecular biology stands as the tribute to human curiosity for seeking to discover and to human creativity, intelligence for devising the complex instruments and elaborate techniques by which these discoveries can be made. So the study of the cell and molecular biology stands as a tribute to human curiosity to seek, okay, for seeking and to discover, and also to human creative intelligence where the complex instruments and elaborate techniques are discovered. Okay, so this is not to imply that cell and molecular biologists have a monopoly on these noble traits. At one end of the scientific spectrum, astronomers are utilizing an orbiting telescope to capture images of primordial galaxies that are so far from Earth they appear to us today as they existed more than 13 billion years ago. So 13 billion years ago, scientists have been capturing the primordial galaxies from the sky. Okay, only few hundred million years after the Big Bang. At the other end of the spectrum, nuclear physicists have recently forced protons to collide with one another at velocities approaching the speed of light, confirming the existence of hypothesized particles, the Higgs boson, that is proposed to endow all other subatomic particles with mass. Clearly, our universe consists of worlds within worlds, all aspects of which make for fascinating study. As will be apparent throughout this book, Cell and Molecular Biology is reductionist, that is, it is based on the view that knowledge of the parts of the whole can explain the character of the whole. When viewed in this way, our feeling for the wonder and the mystery of life may be replaced by the need to explain everything in terms of the working of the machineries of the living system. To the degrees to which this occurs, it is hoped that this loss can be replaced by equally strong appreciation for the beauty and complexity of the mechanisms underlying cellular activity. So this is just an introduction of these cells. Okay, an example of the role of technological innovations in the field of cell biology. This light micrograph shows a cell lying on a microscopic bed of synthetic pores. Flexible pores serve as sensors to measure mechanical forces exerted by the cells. The red stained elements are bundles of the actin filaments within the cell that generate forces during cell locomotion. When the cell moves, it pulls on the attached pores, which report the amount of strain they are experiencing. The cell nucleus is state green. Okay, so that is just an introduction of the first chapter. Okay, now we will be seeing the discovery of the cells. Okay, because of their small size, cells can only be observed with the aid of a microscope, an instrument that provides a magnified image of a tiny object. So if we see the cells, they are small size. Okay, they are in small size. So these cells are observed through only microscope. Okay, so microscope is an instrument which is magnified. Okay, magnified image of a tiny object. Okay, we do not know when humans first discovered the remarkable ability of curved glass surfaces to bend light and form images. So when humans first discovered the remarkable ability of a curved glass surfaces to bend light and form images. Okay, spectacles were first made in Europe in the 13th century, and the first compound double-lens light microscope were constructed by the end of the 16th century. So in Europe, first spectacle was made in 13th century, and the first compound double-lens light microscope were constructed by the end of the 16th century. Okay. By the mid-1600s, a handful of pioneering scientists had used their handmade microscope to uncover a world that would never have been revealed to the naked eye. So the mid-1600s, handful of pioneering scientists used their handmade microscopes to uncover the world, okay, that would never have been revealed to the naked eye. So the scientists have discovered the microscope. Okay. The discovery of cells is generally credited to Robert Hooke, an English microscopist who at age 27 was awarded the position of curator of the Royal Society of London, England's foremost scientific academy. So, discovery of the cells is generally credited to Robert Hooke. Okay, he is an English microscopist. Okay, at the age of 27, he awarded the position of curator of the Royal Society of London, England's foremost scientist academy. Okay. One of the many questions Hooke attempted to answer was why stoppers made of cork, part of the bark of the trees, were so well suited to holding air in the bottle. So many questions were asked for the Robert Hooke, that how Hooke attempted these answers was why the stopper made of cork. So the stoppers which is made up of a cork. Cork means it is a part of a bark of trees. Okay, were so well suited to hold air in bottles. So this cork is holding the bottle tightly so that no air can flow in or flow out. Okay. As he wrote in 1665, I took a good clear piece of cork and with a pen knife sharpened as keen as a razor, I cut a piece of it off and examining it with the microscope. So he wrote in 1665 that he looked a good clear piece of cork and with a pen knife sharpened as keen as a razor, he cutted a piece of it and then examined under a microscope. Okay. Me thought I could perceive it to appear a little porous, much like a honeycomb. So he perceived that it appears like a little pores where it looks like a honeycomb. Okay. Hooke called the pore cells because they reminded him of the cells inhabited by monks living in a monastery. So he founded that these cells looks like a pore. Okay, so these pore cells because they reminded him of the cells inhabited by monks living in a monastery. In actual fact, Hooke had observed the empty cell wall of the dead plant tissue walls that have originally been produced by the living cells they surrounded. So he observed a empty cell wall of a dead plant tissue where walls have been originally been produced by the living cells they surround. So he found a difference between the dead plant tissue and the living cells. Okay. Meanwhile, Anton van Leeuwenhoek, a Dutchman who earned a living selling clothes and buttons, was spending his spare time grinding lens and constructing simple microscopes of remarkable quality. So, another scientist, Anton van Leeuwenhoek, he is a Dutchman where he was selling clothes and buttons. Okay, so he was also spending time for the making of the microscope. Okay, so he spared time for the grinding lens and constructing simple microscopes of a remarkable quality. For 50 years, Leeuwenhoek sent a letter to the Royal Society of London describing his microscope observations along with a rambling discourse on his daily habits and the state of his health. So Leeuwenhoek sent a letter to the Royal Society of London describing his microscope observations along with a rambling discourse of his daily habits and the state of his health. Okay. Leeuwenhoek was the first to examine a drop of pond water under the microscope and to his amazement observe the teeming microscopic animalcules that darted back and forth before his eyes. So he found in the first examine of the drop of a pond water, he found under the microscope animalcules where the darted back and forth before his eyes. So he found animalcules under the microscope. Okay, in a drop of pond water, he found an animalcules. Okay, he was also the first to describe various forms of bacteria which he obtained from water in which pepper have been soaked and from scraping of his teeth. So he was also the first to describe various forms of bacteria which he obtained from water. Okay, so he found some bacteria in water in which pepper had been soaked and formed scraps of his teeth. Okay. His initial letters of the Royal Society describing this previously unseen world were met with such skepticism that the society dispatched its curator Robert Hooke to confirm the observations. Hooke did just that, and Leeuwenhoek was soon a worldwide celebrity, receiving visits in Holland from Peter, the Great of Russia and the Queen of England. So he was another scientist. He was also Leeuwenhoek was also soon became a worldwide celebrity. Okay, he is also receiving visits in Holland from Peter, the Great of Russia and the Queen of England. It wasn't until the 1830s that the widespread importance of cells was realized. In 1838, Matthias Schleden, a German lawyer turned botanist, concluded that despite differences in the structure of various tissues, plants were made of cells and that the plant embryo arose from a single cell. So then there was another scientist in 1838, Matthias Schleden. Was a German lawyer, so he turned into botanist. Okay, so he found despite differences in the structure of the various tissues, plants were made of cells, and that the plant's embryo arose from a single cell. In 1839, Theodor Schwann, a German zoologist and colleague of Schleden's, published a comprehensive report on the cellular basis of animal life. So, in 1839, Theodor scientist, another scientist, a German zoologist, he published a comprehensive report on the cellular basis of an animal life. Okay. Schwann concluded that the cells of plants and animals are similar structures and proposed these two tenets of the cell theory. Schwann concluded that the cells of plants and animals are similar structure and proposed these two tenets of the cell theory. So all organisms are composed of one or more cells. The cell is the structural unit of life. So Schleden and Schwann ideas on the origin of cells proved to be less insightful both agreed the cells could arise from non-cellular materials. So these two scientists, Sladen and Siobhan, their ideas on the origin of cells proved to be less insightful. Okay? They both have agreed that the cell could arise from non-cellular materials. Okay? Given the prominence that these two scientists held in the scientific world, okay, it took a number of years before observations by other biologists were accepted as demonstrating that cells did not arise in this manner any more than organisms arose by spontaneous generation. So they both have founded spontaneous generation. Okay. By 1855, Rudolf Virchow, a German pathologist, had made a convincing case for the third tenet of the cell theory. Cell can arise only by division from a pre-existing cells. So cells have been aroused from a pre-existing cell. So okay friends, thank you. Thanks for watching. If you like my video, please kindly like, share and subscribe it to my channel for more videos. Okay. And please share it to your friends also so that it would be helpful for them also. In the next class, we will be studying about the 1.2 basic properties of cells. Okay, so from here we will be continuing in the next class. Okay. Thank you. Thanks for watching. Bye bye. We will meet in the next class. Bye bye.